Hello, people. What do we got? Oh my God, 22 people. That is roughly 40% of the class. That's sad. That makes me sad. You know what? The cool people showed up, right, Alexa? Right, Bailey? Let's see your faces, people. Come on, let's pretend we're a class for a minute. Emmanuel. Oh, connecting to audio. Emmanuel, let's see your face. Let's bring everybody up. Come on. Farouk. DeJovany. DeJovany. I'm pronouncing your name correctly, aren't I?
No, you're not.
Oh, Jesus. How do you pronounce it?
Di Giovanni.
Jesus. Oh, every time I read your name, I think Di Giovanni, and then I think, no, it's DeJovany.
Yeah,
I have to erase that bit of my brain. Trevor.
Hey.
Diana Johnson shows. Yay, Diana. Oh, and she's gone. Kozlowski. We got a Kozlowski and a Kozlushi. No, Kaczynski. Where's Kaczynski? Is Kaczynski here?
I'm here.
She owes me money. Abigail Kaczynski, Lake Isaacs, and usually you have your camera on. Did you not shower this morning? Who is Kevin? Kevin. Darling Mendoza. Yay, darling. Thanks for coming. Thanks for coming, darling. I'm sorry. I'm sure you get that a lot. Okay. 11. There is Lake Isaacson. Sean Casey. Who the hell is Sean Casey? Looks like he's got a golden retriever.
Golden Doodle.
Golden Doodle. I got a golden. My golden's 120 pounds though.
Oh, jeez.
She's
only like 50 pounds.
Oh,
tiny.
Yeah, I hope my next one's going to be that big. I guess they know how big they are before you buy them, but oh well. Nicole. Yay, Nicole showed. Are you graduating this semester, Nicole? Only if you pass this class. Was that rude? Was that rude? You're doing well. Thaisa Singleton. I see you. Come on up. There she, oh my God. You're living in the dark, Thaisa. Come into the light. Come into the light, Thaisa. Come into the light. Move toward the light. No. That's a totally different thing. Okay, so let's let's go ahead and get started. So all I plan to do today is do a review of the last two chapters because that's what's going to be on Thursday's exam. If you have any questions while I'm going through, it's better if you ask them out loud because sometimes I can't get chats. Like when I'm sharing my PowerPoint and the PowerPoint is open, I can't really get the chats. So either that or maybe we'll assign Bailey to watch the chat box, and if something comes up, she can shout at me. She's been yelling at me for 4 years now, so I'm used to it. Okay. Mitosis and the cell cycle. So I don't typically spend a lot of time on mitosis. Oh, by the way, the new game is up. This figure is prominent in the game. So the new game is up there. Please go and play that when you get a chance. So cell cycle in general. So we got our G1, our S, our G2, and our M. So the whole thing, I don't think I told you this, but the whole thing typically for cells in culture anyway is about 24 hours. So they double on that time scale. Compare that to prokaryotes, which double in 15 minutes, and we're a lot slower. So let's see, DNA synthesis, S phase then is replication. So you're replicating the genome and also checking it for errors. G2 then, the cell grows bigger, so it's ready to divide. Mitosis is the division of the nucleus, really. And then after the division of the nucleus is cytokinesis, which is division of the cell into two cells. Now you've got two cells that are a little bit small. So G1, they grow up to normal size. So that's our cell cycle. Mitosis then, I think what's most important to remember about mitosis is what happens at the beginning of prophase, and then the opposite things happen at telophase. And then in terms of metaphase and anaphase, you know, we get more in detail on that when we get to that checkpoint in metaphase and anaphase. But anyway, early prophase, three things happening. Your cytoskeletons being disassembled and reassembled into the mitotic spindle. And for that reason, the cell rounds up a bit. Number two, the nuclear membrane starts to break down. It needs to get out of the way so we can attach to the chromosomes. And third, the chromosomes condense into this X-shaped structure that we always see on television. So yeah, that's one thing that is starting to bother me. The older I get, the more pet peeves I have. So it bothers me that every time in popular media they show you a chromosome, it's like this, and it's like, well, that's not a functional chromosome. That you only see that in G2 or really mitosis. It's never like that. But anyway, that's our chromosome, two sister chromatids. And then that they attach to the mitotic spindle here in prophase, and then you get this tugging. There's actually, there's actually a lot of physical force going on as they're being tugged in both directions. And as you know, they're attached by these cohesins, so they don't snap apart until you get through that metaphase, anaphase checkpoint. And once they do, you're going to anaphase. We got two things going on here. The chromosomes are going to opposite pole, or sister chromatids are going to opposite poles, and also the cell is stretching out. And that's your microtubules doing all of that. And then telekinesis, the opposite of prophase. So your nuclear membrane is forming, chromosomes are decondensing, and the mitotic spindle is falling apart and turning into the normal interphase cell cycle or cytoskeleton. And then cleavage furrow forms for cytokinesis. And there we are, lovely light microscope view. Here's a scanning electron microscopic view of cytokinesis with the cleavage furrow. So basically there's a belt of actin and myosin around here that contracts down and down and down until it splits into two. In plants, cytokinesis is a little different. You get vesicles full of wall-forming material that fuse together until a new wall and new plasma membrane forms between the two new cells. So mitotic spindle, we've got three different parts to the mitotic spindle. One is the overlapping ones, which are the polar microtubules. Then we've got kinetochore microtubules, and then the astral microtubules. The astral microtubules are pulling these asters to the poles. Remember, before prophase, they duplicate. So now you've got two, but they're next to each other, and they shoot out microtubules that pull them down to the poles. And well, hello, cat butt. And then your kinetochore microtubules attached to either side of to a different sister chromatid, and they tug on it until it breaks apart. And your polar are stretching the cell to make it longer. And there they are. So, and the reason they're doing that is they're connected to molecular motors or motor maps. So these are overlapped by motor maps. You've got motor maps here. You've got motor maps here, pulling this in, pulling this toward it. And then you've got motor maps out here. Leave another picture of that. No, I don't. Motor maps out here connecting this to the cell cortex. Okay. Kinetochore. You know, I've been teaching this for probably 20 years, and I still don't know if I pronounce kinetochore right. I've only read it. I've never heard it said. We're going to call it kinetochore anyway. So anyway, you've got all these proteins that accumulate on the kinetochore. You've got this fibrous corona, as well as these other kinetochore proteins. And this includes things like MAD and BUB proteins that are be important later for that metaphase anaphase transition. And you also have CDC20 here, which is also attached and will stay attached until the chromosomes are attached. Okay, and then cohesins here are connecting the two sister chromatids. And we will have a motor map here as well, pulling this in. But we don't see it. Oh, here we go. So there's your astral attached there. You got motor map here, motor map here, motor map here, and that's where all your emotions are coming from in mitosis. Okay, checkpoints. Checkpoints. So G2 to M transition. This is the first one that was studied, so the first one that we talk about. I think by and large, this one's more important because this is what determines whether the cell is going to divide or not. And losing control of this is really a bad thing. But this is the first one that was studied. So we start out talking about this one. And this one's very different from the other two. So these two are both controlled by cyclins and CDKs. So CDK is a kinase and cyclin is the control molecule that activates the CDK. So CDK then phosphorylates proteins to activate them and that causes things to happen. So cyclins will increase and decrease at different parts of the cell cycle, and that's why they're called cyclins, they cycle. And CDKs are, if the cell is not in G0, you pretty much have CDKs there all the time, waiting for something to happen. Okay, cyclin-dependent kinases are phosphorylating the targets. And we've got four main CDKs. CDK1, which is also called CDK2, is important in that G2 to M transition. CDK2, 4 and 6 are all involved. Well, really 4 and 6 get you up to the restriction point, and 2 will get you through the restriction point. If you take my bio of cancer class next semester, we'll get more detail about that whole process. But for now, just figure these are all involved in getting you through that restriction point. And in terms of cyclins, we've got D and E, which are involved in the R restriction point. B is involved in that other restriction point, the G2 to M restriction point. And A is in the middle of, comes up in the middle of S phase. So again, we'll talk about this more next semester if you take that class, but E comes up, you get into S phase, E goes down, A comes up, you get most of the way through S phase, it comes down, and then B comes up, and then B gets you up through the checkpoint. So, but in this class, all you need to know is these two are important for the R checkpoint. This is important for the G2M checkpoint. Okay, so the cyclins build up slowly. They activate, so we're talking about the G2M checkpoint. When the cyclin and CDK come together and become active, we call it MPF, maturation promoting factor. And again, that's because the early work was done on Xenopus oocytes, and they're like frozen at the after meiosis 1. So this helps them get through meiosis 2 and become an actual egg cell. So that was just the experimental thing that was done to find these cyclins and CDKs. So anyway, I digress. So your cyclin builds up, binds to CDK, a bunch of phosphorylation, dephosphorylation events happen, and MPF activity rises really quickly, gets you through that checkpoint into mitosis. And then MPF actually shuts itself off by phosphorylating its cyclin, and the cyclin gets degraded. And we can see that here. So cyclin's going up slowly, and you get up to a certain threshold, and boom, MPF activates really quickly. And at the peak of MPF activity, cyclin gets broken down. Okay, and that rapid activation is because of these activating and inactivating kinases. So you want to figure through most of this cycle, what's going on is cyclin is being made and it's being phosphorylated to inhibit it, phosphorylating to activate it, and MPF is just in this inactive state for most of that period. And when you get enough of it, just by bulk action, some of it goes through the next step, the phosphatase step that activates it. And then you get this positive feedback cycle that causes it all to activate really quickly. So that's why you get this steep rise in MPF. And then once you get to that peak of MPF, you phosphorylate the cyclin, and the phosphorylation, when cyclin gets phosphorylated, it becomes a target for ubiquitination, gets ubiquitinated and degraded. So, and we've been talking about ubiquitination all semester, and here we see it more directly. You've got specific enzymes, ubiquitinating enzymes, that will add chains of ubiquitin on lysines. Keep in mind, they are lysines, okay? Because that's one of the things I like to do. What if this protein stayed along to around, what could be mutated in cyclin B that makes it stay longer? Maybe it lost a lysine and can't be ubiquitinated. Aha, that's the kind of sneaky thing Dr. Slish likes to ask about. So you add these ubiquitinations, these ubiquitins, which are small protein, onto your protein. That directs it to the proteasome, and then it gets degraded. I just laid a heavy hint there in the last slide. I better shut my mouth. Okay, so what does MPF do? It's a kinase, right? So it's going to phosphorylate things that are going to cause all these things to happen. So nuclear envelope. One of the things we talked about way back in chapter 13 was intermediary filaments. And I said only one thing about intermediary filaments, and that's nuclear lamins that are on the inside of the membrane of the nucleus, keeping it together. So they get phosphorylated and depolymerize, and then the nucleus falls apart, nuclear membrane falls apart. A protein called condensin gets phosphorylated, and that causes chromosome condensation. Mitotic spindle then forms from the activation, I imagine, of microtubule associated proteins regulating the microtubules. And then finally, targeted protein degradation, and that's what's going to shut MPF off by phosphorylating its own cyclin. Okay, so prophase, and then shut itself off. Okay, G2 to M transition. Oh, that's not right. Oh, that's wrong. Sorry. M to A transition. Okay, so this is in that other checkpoint, the metaphase to anaphase transition. We've got all these proteins that are important for it. So let's just talk about the sequence of events rather than defining the protein. So basically, when the kinetochore is forming, you get MAD and BUB proteins associated with it that are also binding up CDC20, so CDC20 can't be released. And when the microtubule is attached to the kinetochore, that releases the MAD and BUB proteins, which then also allows the CDC20 to be released. So, and that's making sure that you don't do anything until all those sister chromatids are attached to the mitotic spindle. So CDC20 then activates anaphase promoting complex. Now, anaphase promoting complex is phosphorylated by MPF, and that kind of primes it to get it ready, but it won't do its activity until CDC20 comes off. And now you've got an active APC, anaphase promoting complex. APC then will ubiquitinate securin, and then securin will get degraded, and that will release separase. And separase then breaks down the cohesins that are holding the sister chromatids together. Et voila. Okay, the chromosomes separate, they go to opposite sides, and you're in anaphase. And the other thing, here we are, so MPF phosphorylates this to activate it, and then this ubiquitinates CDK cyclin, the cyclin, and it gets degraded. Okay. So that's this checkpoint and this checkpoint. And this checkpoint is basically the same, it's just controlled differently. This is going to be controlled by external forces, specifically growth factors. So here is a review of chapter 23. Growth factor binds to receptor, brings up SOS and GRAB2. GRAB2 and SOS. SOS activates RAS. RAS activates RAF. RAF activates MEK. MEK activates MAP kinase. MAP kinase activates June to make it AP1. It also activates ETS. And then you get these immediate early genes being turned on. Now, none of that involves any protein translation. It's all just, all the proteins are there waiting and they all get activated in sequence. Once you have those immediate early genes activated, a lot of them are transcription factors. They're going to come out of the nucleus, or the messenger RNA is coming out of the nucleus. It's going to get translated into protein and then go back into the nucleus and turn on your delay genes, DEGs. So you've got your IEGs come up first, then your DE, I had to look at that for a second. That looks a little bit weird. Then your DEGs come up second. Good thing there's not a third thing coming up, right? Okay, what are the delayed genes? So the delayed genes are the things that are actually going to get you through this restriction point. So the number one is E2F, because E2F is the key to getting into S phase. But as soon as E2F is made, it gets bound up by RB. It's called retinoblast. It's named for the tumor that it came from, retinoblastoma, which is an inherited eye disease where your retina has a tumor in it. That's why it's called RB. So it's a tumor suppressor, and it's a tumor suppressor because it binds to E2F and stops transcription from happening. So when your growth factors, the other things that your growth factors bring up are cyclins and CDKs, especially cyclins. Cyclin D is a really big one that comes up. So then that helps this to get, and then that activation of the cyclin CDK causes hyperphosphorylation of RB, and RB then changes conformation and lets go of E2F, and then the cell goes into that's just rude. That's just rude, Oscar. So then the cell goes into S phase. Okay, so that was for the RAS MAP kinase pathway. But remember, we've got a lot of other growth factors that will, signal transduction pathways that will also activate cell division. And again, I threw this up like I did in chapter 23 to say, look at all these other things that get activated by growth factors. One of them is PI3 kinase. We'll see that in a second. Phospholipase C also can be linked to cell division. I don't know if you remember, but in chapter 24, we talked about 4-ball esters, or crotonoil, which has 4-ball esters, being a growth stimulator. So 4-ball esters activate phospholipase C, so that's how they cause growth stimulation. STATs, JAK-STAT pathway, Shixin adapter protein. So blah, 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 blah. We've got a bunch of other pathways that will activate single transduction too. So here's that PI3 kinase pathway. This is what insulin, this is the insulin pathway, not that important for this example. This is more important. Remember, GSK3 beta is part of that destruction complex that breaks down beta-catenin. Beta-catenin is a transcriptional activator that will cause transcription of cyclin D and cell growth. So the PI3 kinase pathway then causes production of PIP3, which then brings up AKT and PDK1, PDK2, activating AKT, and AKT then phosphorylates GSK3 beta, inhibiting it, which allows beta-catenin to come up in the cell, because when it gets phosphorylated, it gets ubiquitinated and degraded. So this is just another pathway that causes cell division. So will the JAK-STAT pathway, sometimes the TGF-beta pathway, although sometimes it's inhibition of growth. And this, don't worry about this. Ignore this. It's in the book, and that's why I throw it up, but don't memorize it. I don't really care about that pathway. Aha, this pathway is important though. One of the things AKT does is phosphorylate BAD. BAD is a pro-apoptotic BCL2 protein. We'll talk about them in a second, but hopefully at this point you understand the BCL2 story. So if you inhibit whenever the cell gets stimulated to grow, it has to inhibit apoptosis too, because an overstimulated cell will go into apoptosis. So you have to do both at the same time, stimulate growth by producing cyclin D and inhibit apoptosis by phosphorylating BAD. Here is our P53 story. So P53 is called the guardian of the genome because it's going to help protect the cell from DNA damage. So typically P53 is constantly being made, just like beta-catenin, constantly being made, constantly being ubiquitinated and destroyed. So, and again, the whole point of that system is that when P53 is needed, its concentration can double really quickly because you don't have to, transcribe it and translate it. You just have to stop breaking it down. So it comes up really quickly. So anyway, P53 normally is getting ubiquitinated by MDM2, but if there is DNA damage, specifically double-strand or single-strand breaks in the DNA, that will activate ATM and ATR, and that will activate checkpoint kinases, which will phosphorylate P53. And a phosphorylated P53 will not be degraded because it won't be a substrate for MDM2 anymore. So if you've got a little bit of DNA damage, the P53 will bring up P21, which causes the cell to pause in the cell cycle, so it's not going to keep dividing with damaged DNA. And P21 inhibits CDK cyclin, and it will also bring up DNA repair enzymes, which should be on the slide, but isn't. So you transcribe those DNA repair enzymes, you fix the damage, and when the damage is fixed, then P53 starts getting broken down again, and the cell can go back into its cell cycle. Now, if you've got a ton of DNA damage, then P53 will activate PUMA, P53 uprated modulator of apoptosis. I love a protein that tells you what it does. So PUMA then is going to be a pro-apoptotic BCL2, so it inhibits the anti-apoptotic BCL2, and you go into apoptosis. So this is the intrinsic pathway that we'll talk about in a minute. Okay, so that's our P53 story. 3 possible outcomes here. Oh, another interesting thing about P53 that we'll talk about next semester in cancer biology is P53 acts as a tetramer. You can kind of see it's like 4 subunits, so it's a homotetramer. So think about this, you've got two genes for P53, right? So normally you knock out one gene, well, you still got one good gene, so you're probably okay. So if there's a mutation in P53 that causes it not to be transcribed, you still have a good P53 and you'll probably be okay, unless you lose this gene too. But there are certain point mutations of P53 that inactivate it with that, but let it be translated anyway. So then you've got protein being made from both genes, and this is like a dominant negative. So every time it combines with one of these, it shuts it off. So that's the worst kind of mutation to make because it becomes a dominant instead of a recessive mutation. So when P53 is mutated so that it's still produced but isn't going to be isn't going to activate transcription like this, that then that's a really dangerous mutation. But again, the thing we're worried about here with P53 is DNA damage, because once you lose P53, DNA damage doesn't get repaired, gets passed on to the next generation, and that's what produces cancer, more and more DNA mutations building up. We'll talk about that in the next chapter, how you get multiple hits, multiple changes in proto-oncogenes to cause a cell to become cancerous. Once this gets knocked out, that happens a lot faster. Apoptosis. So when apoptosis occurs, what it looks like kind of microscopically, but not on the molecular level, you take a normal cell, which has got a specific shape, and suddenly it loses its shape and rounds up, which immediately says to you, because you took me for cell biology, in your head, you immediately say, oh, cell rounded up, must be a cytoskeleton effect. You're correct. So the cytoskeleton breaks down, the cell rounds up, the cell membrane loses its integrity and starts to bleb out like this. And eventually the whole thing will fall apart into chunks of cell, basically apoptotic bodies. Bleb. So, oh, and also the DNA gets fragmented. That's another part of this project. Cytoskeleton breaks down, DNA fragments, organelles fragment, plasma membrane blebs out, and then phagocytotic cells, macrophages come in and eat up the apoptotic bodies, clean up the mess. Okay, 3, but really 2 different pathways to apoptosis. So the 2 pathways are the extrinsic and the intrinsic, but there's 2 ways to get the intrinsic. So the extrinsic pathway, I just learned something really neat this week from one of the I'll get to it. I'll come back to it. I'll talk about the pathway and then I'll come back to that. So, oh, come on, you. The extrinsic pathway then is when your immune system, your cytotoxic T cells recognize that there's something wrong with the cell. Either it's cancerous or it's infected with bacteria or viruses or something. So it comes in and activates the death receptor on the cytotoxic T cell. And that then activates or brings up FADD, which is a fast-associated death domain. Fast-associated death domain. Yeah, so FAS is the receptor. FADD comes up, and that brings up procaspase 8. Casp8 then gets made from procaspase, and that's an initiator caspase. And that then causes the activation of the executioner caspase, and that activates apoptosis. So you want to think that apoptosis is a good thing if you've got cancer, right? You want those cancer cells to go in apoptosis. Well, someone in the class told me about a drug this week that actually blocks apoptosis. And I saw that, I'm like, what? Why would you block apoptosis? You want apoptosis to happen. Well, it specifically blocks apoptosis in certain white blood cells, ones that are involved in doing this actual process. Because when you've got an infection, those cells come up, but then this apoptosis happens to those cells after the infection's gone to bring them back down. Otherwise, you could have some autoimmune problems. So a new treatment for cancer is to block that decrease in white blood cells so that now they're available to treat the cancer better. Sounds like a really good drug to me. Okay, so anyway, that's your extrinsic pathway. Your intrinsic pathway is activated by 2 different ways. One is the absence of survival factors. So again, going back to, oh God, when did I talk about this? Trophic factors. I can't remember now. But anyway, we talked about trophic factors being important to keeping the cell alive. One trophic factor is integrins binding to extracellular matrix. Another is cadherin junctions, cadherin to cadherin, and that's activating signal transduction. And also growth factor, being exposed to growth factors is a trophic factor. So trophic just means survival. So what all of those things have in common is they are activating signal transduction in the cell. And as I said a few slides ago, that phosphorylates it phosphorylates pro-apoptotic BCL2 proteins and stops the cell from going into apoptosis. So when those things all go away, which would happen if you took a cell out of its normal environment, right now it's not attached to anything, boom, you get rid of the survival factors, your pro-apoptotic BCL2s come up, and they outnumber the anti-apoptotic BCL2s, which causes the release of cytochrome C from the mitochondria and the formation of the wheel of death, the apoptosome, by a combination of cytochrome C, APAF1, and procaspase 9. And then that activates caspase 3, and you're back into the same process you had over here. And then the other way to activate this intrinsic pathway is just by BCL2. So BCL2 comes up, and PUMA is one of these pro-apoptotic BCL2s. Okay, that's apoptosis. Apoptosome. Bye-bye. Okay, that's it. That's it for chapter 24. See, I could have did that whole chapter in half an hour and everyone would have known just as much as they do now. Here's the share line. Am I really? Stop. Let's do a new share. Okay, chapter 26. Cancer biology. So, starts out with the basic definitions of benign versus malignant. And remember, the key is always this basal lamina, which, and the reason we say that is because most cancers, like 90+ cancers, are epithelial in origin. And the epithelial layer of cells is separated from the inside of your cells by this basal lamina. Now, there are other tumors that we'll talk about in the class in the fall, but for most of our tumors are going to be epithelial. So this basal lamina is what's making the difference here. So your benign tumors will stay within the epithelial layer. Malignant will invade into surrounding tissues. And then your metastatic then are the ones that invade and then go into blood vessels or lymph vessels and move to other sites in the body and set up new tumors. Okay, benign, malignant, and metastatic. Okay, and then, you know, and that's when they become metastatic, that's when you usually die, because usually a tumor in your you know, epithelial tissue is not really going to kill you, but once they go into your bloodstream, usually they'll set up new tumors in the lungs, in the liver, in other tissues that are vital for life, and that's what usually kills you. And that's because, you know, when they get into the bloodstream, you know, they're going to get into the venous system. The venous system comes back to the heart, and the vessels keep getting bigger and bigger and bigger. So the opportunity for it to hit the wall and get out is really bad. And also the speed of that blood is going faster. So it's really hard for them to get out. But then they go through the heart, and then they go to the lungs, and the vessels get smaller, 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 and go slower, 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 and then they sneak out. So that's why you get it there. It's like the first capillary bed that they see. And also they'll set up in the lymph nodes, because if they go into the lymph system, that is the first system that they will see is the lymph nodes. Cellular differentiation. So here we start talking about what makes a cancer cell different from a normal cell. And one of the big things is dedifferentiation. So differentiation is the process of going from a general kind of embryonic cell to an adult functional cell. So these cells back here are not really functional. They don't perform any kind of immune function. They're just on their way to becoming that immune cell. So cancer cells will dedifferentiate. They'll go back into this progenitor type, more embryonic type cell, which means they're going to divide and not perform a function. So you get a whole lot of cells that are just clogging things up. And here we are talking about differentiation again. Here's your basal lamina. Here's your epithelial stem cell. They divide, one goes up, one stays here to be a stem cell again, and they continue to differentiate as they go towards the surface. And this differentiation in the skin is basically filling the cell up with keratin, so it's full of protein, and that forms that nice waterproof surface that allows us to live outside of the water environment. And when you get tumor growth, they're not differentiating anymore. They're staying pretty embryonic and growing faster. Now, when they break through here, they become malignant. All right. Another thing that they have is self-sufficient growth signals. And so they're going to replace that external need for trophic factors with just an internal mutation that gives them the trophic factor they need. So you could have an activated AKT or an activated focal adhesion kinase or PI3 kinase, really any of these kinases up here that are involved in causing the cell to avoid apoptosis could be activated. And then the cell can break free of its extracellular medium and not need growth factors anymore to survive. So that's called anchorage independent growth. Typically they're anchorage dependent and cancer cells become anchorage independent. And also with those cadherin attachments, that causes cells to stop growing when the tissue is all put together, and now we've got enough cells you can stop growing now, and that is density dependent inhibition. So that's another thing that cancer cells lose. They lose their density dependent inhibition. So even though they're already in a tissue and everything's okay, they keep dividing anyway, and then that's how the tumor forms. But I'll come back to this. They can only form, so they can only get, that tumor can only get so big before it needs to get a blood supply. Because when it gets to be about 2 centimeters, this is 2 centimeters, right? More or less. When it gets to be about 2 centimeters, the cells in the middle start to die because oxygen can't diffuse all the way in there. And so they're kind of stuck at that point until they start putting out hormones like VEGF. What else? VEGF, maybe FGF, also endothelial growth factor, something like that. And that will cause the angiogenesis to happen. So bringing a blood supply to itself. The other thing it needs in order to keep dividing is telomerase. Telomerase is going to extend the 3' end by putting these repeats on the end of it, because during replication of a linear chromosome, the chromosome keeps getting shorter, shorter, shorter. So without telomerase, the cell will eventually die, but with telomerase, it can be immortal. So telomerase is another important thing for cancer cells to have. Self-sufficient growth signals. So we'll talk later about proto-oncogenes versus oncogenes. Everything, the genes that make anything in this pathway will be a proto-oncogene, okay? Because if it gets mutated, so it's out of control, it's an oncogene. So any one of these proteins in this pathway, if the cell loses control of it, either by overexpressing or point mutations making them constitutively active, that converts that proto-oncogene into an oncogene. So cancer cells are self-sufficient in terms of growth signals because they've got oncogenes that are replacing these normal cells, and they keep dividing because of that. Okay, how does a cell go from benign to metastatic? So from benign to malignant, how does it start invading tissues? It's got 3 processes to go through. It has to let go, clear a path, and crawl. So letting go involves changes in cadherins and changes in integrins. So the cadherins are, causing density-dependent inhibition and also keeping those cells in the tissue. So when they let go of that, now they can start moving away from that tissue. And the integrins then allow the cell to crawl out of that position and move somewhere else. But of course, there's nowhere to go in a tissue because everything's all together. There's extracellular proteins over there, the extracellular matrix keeping it all in one spot. So you need to get rid of those proteins. You have to get rid of that extracellular matrix. And the way that happens is by releasing plasminogen activator. And plasminogen activator that will activate plasminogen to make it into plasmin. And plasmin then will cause the activation of matrix metalloproteinases. And they will go out and break down that extracellular matrix. So listen to that pathway. You've got the cell will release plasminogen activator, which activates plasmin, which activates metalloproteinases, and that then destroys the extracellular matrix. So that's clearing a path. And the last thing is increased motility or crawling away. And we talked about this in chapter 14. The cell puts out a lamellipodium, makes new focal adhesions, and then the stress fibers pull the cell forward into that lamellipodium. Basal lamina, we're back to the basal lamina. So your matrix metalloproteinases will break down that basal lamina and allow the cell to invade. What causes DNA damage to give you cancer? So one thing is carcinogens, chemicals that mutate your DNA. Cigarettes, I always focus on because it's the most obvious thing. A lot of carcinogens in cigarettes. Your liver can activate different chemicals to make them carcinogenic. And then of carcinogens, you've got things that cause DNA damage, and then things that will promote those damaged cells to become cancerous. So you've got your initiators and your promoters. So the promoter alone doesn't do much. The initiator alone doesn't do much. Together, they make sweet, sweet music. No, they make cancer, sorry. So croton oil then has got 4-vol esters in it, which activates protein kinase C. I talked about that earlier today. Okay, ionizing radiation can cause DNA damage. So usually we talk about UV because your skin cells can be damaged by UV. Also, this is why, being in a nuclear blast is a bad thing. It's a bad thing. I would avoid it at all costs because gamma radiation will also cause DNA damage. Beta will too, but they don't invade your skin very far. If you swallow something that's a beta emitter, then you can get cancer from that. What else? X-rays, you don't want to get too many of those. Do you ever go to the dentist and they put that big lead thing on you? Oh, I just want to stay the whole rest of the day. It feels so comfortable with that big lead apron on me. I guess I have to get a weighted blanket. So anyway, ionizing radiation can cause it too. Viruses. There are not a lot of viral diseases. Warts are a viral disease. This is herpes is another viral disease, similar to warts, I think. So HPV is a viral infection, and it can cause DNA damage. And that's because, oh no, it doesn't cause DNA damage. It causes cancer because in the genome of the HPV is some oncogenes that used to be proto-oncogenes, but the virus grabbed it out of someone's genome, and now it's part of the virus, and now it gets replicated and passed on. Okay, stepwise development of cancer. So making one proto-oncogene into an oncogene will probably not make that cell cancerous because the cell will have different, the body's got mechanisms, the cell's got mechanisms to kill bad cells, so that would get taken away. But if you have if the cells are allowed to keep living and dividing, they get more and more DNA damage in them, and that causes them to become cancerous. So probably a good example here is in colon cancer. So this is the surface of your colon. This is where the poop is. So these cells are constantly being replaced from these cells in the crypt. At the bottom of the crypt, you've got stem cells that divide, and cells, after they divide, will differentiate until they reach the surface, and then they'll die and constantly being replaced. So if you get mutations in these cells, that's what will then cause them to become cancer. So this is a polyp. Again, not dangerous. The doctor can come in and rip all this out and get rid of it. But you get enough mutations, now you've crossed that basal lamina and it's invading. So, and the point of this slide is you get one mutation, it gets a little bit overactive. You get more mutations, it gets more overactive, and more mutations, then it starts to invade and become really dangerous. So again, it's like, it's not one mutation that causes it, but multiple mutations in the same cell that makes it more and more dangerous. So we'll talk later about what you need to do. So, okay, so here's just APC is really important here. So APC is part of the death complex that breaks down beta-catenin. Without APC, beta-catenin just builds up even when the cell's not being stimulated. Adenomatous polyposis coli. Okay, how do genes become, go from a normal proto-oncogene into an oncogene? One way is by point mutation, and that mutation has to be in the right spot. So most mutations are not going to change a protein much, but if it's in the right spot, like in the GTPase pocket of RAS, okay, you knock out the GTPase, now the protein is constitutively active. So point mutations can make cells active. Gene amplification. So sometimes, well, if you amplify that gene, you'll get more gene product, right? So you have more protein. So excess of the normal protein can cause a cell to be, can cause the proto-oncogene to become an oncogene. Translocation. So this is when you start having those double-stranded breaks in DNA that are not being fixed. So the strands break and then they attach to other chromosomes and you get translocations happening. So one example of a translocation is a chunk of one chromosome switching with another chromosome. And that can put something important like MYC, which is it's an immediate early gene that causes cell division, put it from a quiet place in chromosome 8 into an active region of chromosome 14, and now you're getting MYC when it shouldn't be made, and then the cell becomes cancerous. You could also make a whole new protein by a translocation. So that's this BCR-ABL protein, which is made from the BCR protein and ABL protein. And now it's an unregulated kinase, and it causes cells to become, that gives it its, growth signal. So it's self-sufficient for growth signals. Another thing, local DNA arrangement, so not between chromosomes, but within one chromosome, you get an insertion or deletion, which can knock out no, we're talking about proto-oncogenes. So that could cause abnormal or hyperactive proteins. Here's a deleted piece. So that can cause activation. Here is a paracentric inversion. Paracentric around the center. So it breaks here and here, flips over, reinserts. So this is an interesting one here where you get combination of a NGF, a nerve growth factor receptor, with tropomyosin. It clips out, flips over, flips back in. Now you've got a new protein, and that's the TRAC oncogene, which has got tropomyosin, making sure that these receptor domains are always stuck to each other, and you have a constitutively active growth factor receptor now. And then viruses can cause cancers as well, even if the cancer itself isn't a cancer virus like HPV is. Like any other retrovirus can insert into a genome and cause the next door neighbor proteins to start being made. So, if you'd have a virus that sat down next to MYC, MYC would start being made, and that would make that cell cancerous. And this is your retrovirus. So, blah, 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 blah. Okay, so again, like I said before, anything along that growth factor pathway, if it's mutated in some way, could would convert that from a proto-oncogene into an oncogene. So growth factors are made from proto-oncogenes. If you overexpress the growth factor, cells got self-sufficient growth signals. The receptors could be mutated, the G proteins, kinases, transcription factors, cell cycle regulators, all these things are made from proto-oncogenes. And if you lose control of that proto-oncogene, it becomes an oncogene. And the other half of the story is the gatekeepers, the tumor suppressors. So these are proteins that stop the cell from dividing. So probably the most obvious one is RB, right? Because RB is holding E2F. So if you don't have RB anymore, E2F, as soon as it gets made, the cell starts to divide. There's no control anymore. APC, right, is activating GSK3 beta and blocking cell division. P10 is converting PIP3 back into PIP2, and that's turning off that pathway. So all of these things are proteins that will block cell division. And if you lose it, the cell is more likely to keep dividing. So those are gatekeepers. Then you also have caretakers, which are enzymes that fix broken DNA. So if you lose those, then mutations just start to build up because you're no longer repairing your DNA. So those are the caretakers. And that xeroderma pigmentosum is one example of that. Okay, so just a quick review here, summary and review. Hallmarks of cancer, they're self-sufficient in their growth signals. They're insensitive to anti-growth signals, evade apoptosis. They've got telomerase, so they're immortal. They have sustained angiogenesis, and they invade and metastasize. Bum, bum, bum. See, I could have done these two PowerPoints like that, like weeks ago, and we could have had all these days off, I could do this whole class in I don't know, 3-4 sessions, and we can take the rest of the semester off. I should try that next year. Do you feel like you learned anything today? Thank you, Claire. Any questions? Comments? Nothing. Carrie Benatti always has questions. She's afraid to ask them, though. Graylin Larch is always quiet. Farouk knows all the answers, so he's not going to ask. Trevor thinks he knows all the answers.
I think you did a great job today, Slish.
Thank you.
It's all crystal clear.
Okay, so that's all I have. So shall we call it a day? Exam on Thursday. There will be some multiple choice or objective ones anyway, and some essays as usual. And then the final next week will just be the objective ones. Okay, everybody happy? All right, we'll see you guys on Thursday. Take care of yourselves and each other.